All right, so we're still continuing on in chapter 33, finishing up the insects, and we'll also in chapter 33 talk about the next group of organisms, which actually ties into the very last chapter we'll go over, which are the deuterostomes. But we'll get to those in just a minute. What if you haven't, which you may have, if you haven't really sort of grasped what I'm trying to get at with the insects through this discussion, the biggest thing about the insects I think I can say, or excuse me, let me back up for a second. It's certainly true about the insects, but the arthropods is the diversity. This is an extremely diverse group, and that diversity is probably no more better illustrated than among the different species of insects that are out there. As I said, somewhere in the vicinity of over 30 orders of insect alone, which we're not going to look at all of them, but that means an extremely, what we call a speciose organism. Speciose means lots of species. O-U-S. There we go. A speciose organism, so arthropods would be very speciose. Not only that, they're also very heavy in density. Remember, what did we say, 10 to the 18th arthropods are live at any one time. And their diversity has a lot to come from the many different ways they can exploit their environment. And that's kind of what we were working through and talking about some of these adaptations. And one of the adaptations that they use is the different mouth parts, depending on what kind of critter they are. So you see on the right-hand side, if you're something like a mosquito or a tick, many times a blood parasite or a blood feeder, you're going to have a piercing proboscis. Proboscis is a fancy name for a nose, I guess. Proboscis, I think that's how it's spelled. So the proboscis on the mosquito will be piercing. Butterflies, since of course are suck the nectar out of the flowers, they want that really long tongue so they can get very far back into that flower to be able to get all that nectar. And then our buddy the fly has a very interesting way, has a very interesting modification to its mouth part. When we're outside and we're having lunch or something like that and a fly flies on your food and you quickly shoo it away, right? No big deal. But what you may not be aware of is when that fly, for the period of time that it was walking around on your hamburger, was actually regurgitating onto your hamburger, using that regurgitant as sort of like the fungus do with exozymes, using that regurgitant to start the digestive process of, in this case, your hamburger, and then they sop it up. So they basically, sopping is to like to lick up. So like a bird, like a dog would, sop up, say, gravy out of a bowl. So that's kind of interesting when you think about that fly. And then as it's crawling around, it's actually kind of puking on your food. And of course, you shoo it away, bye fly, and you eat the hamburger without ever thinking about it. But there's a very good chance if you're outside and there's flies around and you're eating, you probably have or will have or have had at some time in your past eaten a little bit of fly regurgitant. Sounds wonderful, doesn't it? I think there's even that provision in manufacturing of foods, that certain foods, is it candy bars or something like that, can have X amount of insect parts in them. They're allowed a certain amount of insect parts before it's considered well, hell, I don't know, before it's considered gross, or before it's considered releasable to the public. I want to say like 10. I might be wrong. Not 10 full insects, but part of a leg or something like that. Excretion in these guys, excretion in the arthropods is going to work the same way that we talked about in the grasshoppers when we were discussing the Malpighian tubules. And again, Malpighian tubules were very much like the flame cells that we saw back in the flatworms, and to some degree that we saw in the segmented worms, where you have that one cell with that cilia that's beating and that's filtering water into the tubes. Now, in the Malpighian tubules, there's not the cilia, but through diffusion and osmosis, these things diffuse into those Malpighian tubules. There's a new commercial on TV, and I wanted to try to remember it, and I'm glad I did, because it's for, oh, crud. Anyway, part of the commercial says something like, according to scientists or something like that, the wings of a bee are too small for a bee to fly. So it's that old adage, and maybe you've heard it before, that the bees defy gravity, defy physics. They shouldn't be able to fly because their wings are too small for their body. Well, whenever I hear that commercial, I'm reminded to talk to my students about that. So if you have someone who said that, and I've heard this more than once, It's always interesting to me how folks come up with this idea that there's magic out there still, right? You know how bees fly with wings? No, 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 no. With their wings, it would be impossible for them to fly. Well, how do they fly? Well, we don't know. Well, yes, we do. Their body is big, yes, compared to their wings, but a lot of flight insects, a lot of insects with flights have taken those trachea and those spiracles. Remember those tubes that bring oxygen into the insect's body that I showed you with the grasshopper a few slides ago. In flighted insects, like bees, for example, those tracheas can actually be expanded into air sacs. So while the bee on the outside looks too big for the wings, you know that, of course, with those air sacs, what we got is a large, okay, but a very light organism, a very light insect here. So it is not impossible for bees to fly. It really has little to do with the size of their wings when you factor in that if you look at that picture on the right, you know, with the bees and showing the air sacs, that quite a bit of the body on the inside is full of air sacs. Now, this is something that I think is really fascinating, especially when we do the comparison to the vertebrate flyers, the birds. Birds have this too. Birds have geez, Steve, what's wrong with you today? Their bones are hollow. You lighten the load, which makes it easier to fly. So no, of course not. It's not. My answer to that with people is always, do bees fly? Well, yes. Well, then it's not impossible. What do you mean? Oh, well, you just told me if it's impossible for bees to fly, but quite clearly they do. So it's not impossible. All right. In some organisms, and this is something where we kind of do the detective work again. So you're studying an insect and you notice that the spiracles on an insect are all closed off, like they're not open to the outside. You might have either a parasitic or perhaps an aquatic insect, an insect that wouldn't want materials moving in through the spiracles. If you're parasitic, many times organisms that are parasitic will actually be able to exchange gases through their body wall, so they don't really use the spiracles. Diffusion is the process by which it's done, though. Regardless of whether you have big air sacs or just regular spiracles and trachea, the air diffuses in, and then the oxygen diffuses across the cell membranes into the cell. Remember, in diffusion, you go from higher to lower. Inside the cells, the oxygen is being used. So in the tracheals that are outside the cells, right, it's going to diffuse into it. The tympanum, I mentioned when we talked about spiracles. There's a picture in the lower right of the white arrow is pointing to the tympanum on a butterfly. Yeah, that's a butterfly, Steve. A grasshopper. Tympanum it's like a drum head. It's a very thin membrane that's been stretched over some spiracles associated oftentimes with these tracheal air sacs. And of course, they use that for sound reception. Now, is there sound reception again like ours now? Probably not. It's more of a like in some organisms with their light can see shadows. You hear sounds kind of maybe at that resolution. Many insects, of course, in the middle picture are the ants, have pheromones, okay? And pheromones are an interesting chemical. Pheromones, think about external hormones. Pheromones are chemicals released into the environment by an organism that transmits a message. It could be a mating signal. Many species of insects, the females will release a pheromone into the environment. When it's time to mate, males will smell that pheromone and find a mate. It can be used for trail markers. Ants, as ants are moving along, you know, they always seem to walk along the same trail. Like if you got ants crawling in your house and you mash on them, you know, and then a few minutes later, there they are again, walking exactly the same trail. It's a little like a Hansel and Gretel routine. What those ants are doing when they're walking is they're dropping a little bit of these pheromones, these chemicals, so they're marking their trail. That's how even when you kill them all off, they know exactly where to go, you know, where the same exact trail. The treatment for that, by the way, they say is when you kill off the ants or whatever, get rid of the ants, you have to use something like, say, bleach or some other solvent that would actually get the pheromones, clean the pheromones off, because they're still there, even if the ants are gone. Oh, I missed CD up at the top. CD are hair-like structures, okay? Hair-like because hair, again, is only in mammals. And these can detect chemical messages. They can be chemosensory, mechanical, brushing up against it. seedy or these little hair things can also vibrate. And here's a really good picture on the bottom. I think this is on a bee. I don't remember exactly what, but all these little fuzzy things sticking off the bee are seedy. So chemical, mechanical, and sound signals. The mosquito larvae down there, the reason why I'm showing you that, it actually has to do with the breathing and the spiracles. Mosquito larvae, of course, mosquitoes lay their eggs in the water and they hatch in the water. And the mosquito larvae actually develops in the water for a while. And what it does is it hangs right down below the surface of the water. And it's got this little device that extends up to the surface. And that's actually a pipe. And that's how mosquito larvae get oxygen, exchange gases while they're developing as a larvae. Reason why I bring that up is one of the earliest techniques for getting rid of mosquitoes was to place oil over the top of the water where the mosquitoes were nesting or were reproducing. The idea behind the oil, of course, is it plugs up the feet or the air tube of the mosquito larvae and kills the mosquito. Dumping oil into natural waterways is a really, really, really bad idea, by the way, because while indeed may take care of the mosquitoes, all of the other stuff that's in oil is really bad for the organisms living in the water. So not a good way to treat mosquitoes, and of course there are a lot more effective ways of doing it now. Maybe with things that are actually better or worse for the environment in the case of pesticides. Most insects undergo sexual reproduction. Since so many of them are land dwellers, the fact that they have internal fertilization should not be surprising. That's kind of the norm. There's so many of them and so much to eat that one of the things that insects came upon, this really unique adaptation and idea, was this idea of a metamorphic state. All right, so they undergo, especially in the case of the butterfly, they undergo what we call a complete metamorphosis. A complete metamorphosis means that the larvae looks nothing like the adult, right? And this is something we've known since grade school. Butterflies and moths come from caterpillars, right? Caterpillars, the larvae that erupts out of the pupae or the chrysalis, and then the larvae, caterpillar feeds for a while, and then eventually, or the caterpillar, then it forms the chrysalis and forms the butterfly. Got that backwards a little bit there, sorry. So that would be complete metamorphosis. Again, the reason why that would be something interesting about insects is one of the reasons we believe that this complete metamorphosis may have evolved in certain organisms is because it reduces competition, right? We talked about dispersal is the whole idea of getting your young, go, get away from me. I don't want to compete for resources. But another way that can be done, in addition to that dispersal, is you can make the young different, and they feed on something different. So the caterpillars feed on one part, and then of course once they metamorphose in the chrysalis into a butterfly, then the foodstuffs typically tend to be something different. So that's kind of an interesting way of dividing up the resources in an environment. It's sort of an interesting approach to this solution of limited resources that Darwin talked about way back at the beginning of the semester, right? This whole idea of limited resources. This is a great way to get around that. Now, other insects, of course, grasshoppers, for example, have what we call incomplete metamorphosis. Sometimes it's called simple. And on the left, you can see the two insects that I'm pointing. I think that's a silverfish, if I remember right. Those are little insects that crawl all over the place in your closets all the time. Anyway, the younger ones are just smaller versions of the adult. So that's incomplete metamorphosis. Okay, so that's all I want to say about the insects as far as their characteristics. I want to move in now and talk or the arthropods, excuse me. I keep saying insects because they're one of the biggest groups. I now want to move into some of the groups. Again, 30-something orders. I'm not going to expect you to know each of these. The ones in red that I go through, those are the ones that I really would like you to concentrate. So first of all, we look at the winged insects. There are many orders. There's only, a half dozen here. The biggest group is going to be the beetles, the Coleoptera, followed right behind that by the Diptera. Now, what I've done is I've tried to put little hints in there to help remember what group these belong to. So the insects with sheathed wing, all right, is called, a sheath is a coleus. So these would be the insects that have it looks like two sets of wings. Basically, the one thing comes up and then the wings are underneath it. Those would be beetles. Coleoptera. Diptera only have two wings, so they don't have the beetle wings. The first wing, the sheath wing, the sheath over the wing, it's actually not a flyable structure. The wings are underneath it, but the Diptera organisms you see, like the I don't know what the kind of little insect that is. It's not a bee, but they've only got two wings. Hymenoptera. A hymen is a very delicate membrane, very thin membrane. And if you remember, if you've ever seen the wings on a bee, they are very thin, aren't they? Right. So Hymenoptera. And I remember Hymenoptera bees because you have to kind of follow me the associative learning. Hymen is the tissue that is ruptured the first time a female engages in intercourse, and then bees have our Hymenoptera, so I put birds and the bees together, and I remember, okay, Hymenoptera. I'm sorry, that's kind of a around your elbow way to try to remember something, but I'm just giving you ideas of the way I try to remember these things. Lepidoptera. Lepid is scaly, so I think of lepid as lepidosaurs, lizards. Okay, so scaly-winged Lepidoptera, those are the butterflies. The Hemiptera, they have what we call half wings. It's not really half a wing, but they have two types of wing. One wing is textured, like you can see in the picture there, and then the other wing is non-textured. The Orthopoda orthodox, they have strong, or not strong, but very straight legs. The Orthopoda tend to be, for the most part, some of the more ancient types of insects. Now, the ones on the left, the beetles and the bees and all that, undergo complete metamorphosis, and the ones on the right, then again, undergo incomplete. So the Coleoptera, that's a cool-looking guy, isn't it? The male snout weevil. See, his proboscis has obviously been modified for some sort of insertion into something, probably into flowers or somewhere to get his nutrients. It looks a little bit too big to be piercing. Oh, the Diptera, yeah, mosquitoes. That's who belongs in this group. I always remember that dip. We use a dip to refer to someone we don't particularly like. So mosquitoes are something we don't particularly like. There's our buddy the mosquito, and he or she is round up on its rear legs, and there's the proboscis that's digging down into your skin. I read a really interesting article the other day that oftentimes when you get bit by a mosquito, you don't know it until afterwards, right? You get the itchy. And you would think, if something is punching a hole in me with a proboscis, like a little needle, I would feel it. And sometimes you do, don't get me wrong, but oftentimes you don't. So when biologists begin to look at the microscope structure of the proboscis of a mosquito, they found something very interesting. At the very microscopic level, the proboscis, or the needle part of the mosquito, is not straight. It's not smooth. That it's actually, and again, I apologize for my lack of artistic ability, it's slightly serrated on the edges. It's almost like a serrated knife. Now, the serrations aren't this extensive, but those serrations, we believe, actually diminish the sensory input of the organism who is the host. So what I'm saying is, because the proboscis of the mosquito is designed like that, I guess it should be more to a tip down here, because it has these slight serrations to it, it actually benefits the mosquito, because the host, it's more difficult for the host to feel that proboscis being inserted into him. The actual point of the article, though, was interesting, because I always love this idea where we see in nature how nature's done something. We go, oh, I wonder if we could apply that here. Designing immunization needles to not hurt, right? Shots, when you get shots, one of the ways they're thinking about it, if they design them in a way with these microscopic serrations, kind of like the mosquito does, they wonder if they would make immunization needles that wouldn't be nearly as painful. That red taconid there, he looks pretty vicious, doesn't he? Looks like he means business. Of course, paper wasps. There's European paper wasps. We have paper wasps. Paper wasps are those ones that make those nests that basically are cellulose fibers that they chewed up and turned into these very nice, regularly shaped nests. About 125 thousand species of them, about 120,000 species of the moths, butterflies, or this really cool thing called a hummingbird hawk moth. That's a big moth, right? It's almost hummingbird. It's almost bird-like, and it's obviously in this case got its food through the nectar out of the flower, so it's got a nice long extendable proboscis. Oh, the good old stink bugs. This is the adult stink bug with a lot of females, with a lot of offspring. Saw a really cool spider today out in the hallway in the biology building, and I got down close enough, and she was actually carrying eggs on her abdomen. One of the reasons that female arthropods, crabs as well, we talked about this, have larger, more extensive abdomens is because that's where they often carry the young. The eggs of their young are on their abdomen. And then this guy, this is a katydid. That's what this is a katy, the common name. We have katydids here in Florida. I don't know if we have this one. This is the spear bear. I spelled it for you. That's right there. That's a cool looking critter though, isn't it? Okay, just a couple of other smaller groups. For example, my favorite, I have to introduce the dragonflies, the Odonata. Odonata odontis, it means teeth. Dragonflies have teeth. Some of the most ancient critters that have been around, dragonflies pretty much look the same way they do now that they did even back during the dinosaurs. So it's kind of like a living fossil. The termites, which of course are problematic here in Florida because they like to eat our houses. Termites belong in the group called Isoptera. Iso means equal. So they have 2 pairs of wings, or they're wingless, and the wings are of equal size. That's what it means by that. Termites are really strange, too, because they're a little like bees in the sense that they build these elaborate social organizations. And, of course, our buddy the flea, the dog flea, Siphonoptera. The Siphonoptera comes from the fact that it's a blood feeder, right? It uses its siphon. So proboscis right there. And they don't have wings. So Aptera means without wings. So Siphonoptera. And those are ectoparasites is what we call them, right? Parasites that live on the outside of organisms. Get their meal that way through the blood. Well, that's it for the arthropods. That's it for most of the traditional types of invertebrates. We're now going to move into one more group of invertebrates, but we're going to be moving into a larger group that those invertebrates are contained with, called the Deuterostomia, or the deuterostomes. Remember, the deuterostomes has to do with embryonic development. Deuterostomes and protostomes. All of these here are the protostomes, right? And then the deuterostomes are the ones in which the anus forms first and then the mouth forms. Now, there's 3 groups in that we'll talk about. A small group, the Hemichordata. We won't really talk about them too much. The echinoderms, which is what we'll talk about today, that's our last true invertebrate. And then in this group is mixed in the rest of us, the chordates. Chordates, literally, the word chordata comes from the Latin that means string. So we're talking about those organisms that have a vertebral column. Now, that's going to become a little bit confusing when we get into the chordates because, let me say this the correct way. All organisms with a backbone, okay, all members with a spinal column are chordates, okay? Not all chordates have a spinal column, all right? So you'll see what I mean in just a second by that. But let's finish off with the last invertebrate. Actually, if you look at this chart here, it's invertebrate that us chordates are the most closely related to. I mentioned a couple times in class that when they were trying to ascertain which invertebrate group may have given rise to the chordates, some of the early candidates were the segmented worms, the arthropods. I think there was another one. It might have just been the segmented worms and the arthropods. But come to find out, for reasons that we'll get into in just a moment, it actually is the echinoderms. All right. The word echinodermata means spiny skin. So we're going to be talking about spiny skin creatures. So there we go, deuterostomes. Deuterostomes are everything from you and me, the white shark in the upper left, that rhino on the right, to all these weird little things. This is a sea cucumber, very odd looking critter, but you are distantly related to that. You're more related to that sea cucumber than you are to insects, right? Or other invertebrates. And then, of course, sea stars. Sea stars are very typical with 5 arms. They don't have to have 5 arms. This is a Florida species of sea star. I can't remember what it's called. I think it's something called very plain, like the brown sea star or something like that. All right, but the deuterostomes. All right, so about 670 million years ago, we get this bilateral group that breaks off into the protostomes and then into the deuterostomes. And again, like I said, what we're going to be talking about specifically first in the deuterostomes are the spiny skin guys, the echinoderm, spiny skin. All right, so these are the sand dollars, the sea biscuits, sand dollar, sea biscuit. Sand dollars are flatter, sea biscuits are thicker. The sea urchins, there's a sea urchin. Sea urchins have a test. Remember, that's the word we use to refer to the shell and diatoms, and dinoflagellates. In the foraminiferae, the test was made of calcium carbonate. This is the test of a sea urchin. So in other words, if you pulled all the spines off of a sea urchin, that's what you would see. You would see this ball, this calcium carbonate ball. Okay. Echinoderms are exclusively found in the ocean. They exhibit a type of symmetry, a special type of radial symmetry we call pentaradial, oftentimes pentaradial, which means 5 parts to it. And again, sea stars very often have 5 parts to them. So here's the interesting thing about it, and here's where this confusion that you might have about relationships. Echinoderms and chordates like you and I are the most closely related as far as invertebrates go. We have bilateral symmetry. We've been dealing with organisms that have bilateral symmetry since back we've been talking about the worms, the flatworms, the roundworms, the segmented worms, and of course all the mollusks, well, many of the mollusks, and the insects and the arthropods we talked about had bilateral symmetry, left and right. So if I've talked to you before about, similarity in embryonic development is very often indication of relationship or close relationship. So if you both exhibit radial symmetry throughout your embryonic development into adulthood, it may be a indication of relationship. Or like the cnidarians, the jellyfish and the comb jellies, right? They both have radial symmetry and among many other things, and that actually is relatively small part that indicates there's some relationship between them. But we don't have radial symmetry. It is still about the embryo, though. It's still about embryonic development. If you look up here on the right, this is the larvae of an echinoderm. So this is the larval form that eventually metamorphoses into a sea biscuit, a sand dollar. Oh, sea pen. That's a sea pen, P-E-N. I guess like the quills people used to write with me. That's where it came from. Sea cucumber. The knife is there for scale, folks. It's not holding it in place. Obviously, we got radial symmetry here. We got radial symmetry here. Don't really have radial symmetry there at all. But if we look at the larvae of the echinoderms, the larvae of echinoderms is actually bilaterally symmetrical. So it's got a front and a back, a left and a right, top and a bottom. So when we get into the chordate chapter, I'm going to start about right here talking about where chordates came from. Because what we think happened is we think that a larval echinoderm, all right, a bilateral, larval echinoderm underwent some changes. Let me get paedomorphosis, patagenesis, neoteny. You remember those words back in evolution whereby an organism looks like a juvenile, but actually develops the ability to reproduce. We say that what goes on in those processes, again, remember, is that somatic cells, which are your regular cells, and reproductive cells are developing as you're developing. And in this paedomorphosis, neoteny, patagenesis thing, what happens is the reproductive cells mature faster. So you end up looking like a juvenile, but you're reproductively mature, so you are an adult. So that's what we think happened here with echinoderms. It's very, very cool. We think there was some bilateral echinoderm larvae that through metamorphosis and change and evolutionary adaptation eventually gave rise to the chordate. So that's why, in spite of the fact that when we look at echinoderms and we think, wow, I would have not picked that critter to be our closest relative among the invertebrates. Indeed it is. Remember, going back to that embryonic development. Not much of a nervous system, not really a brain in these guys either. They have a nerve ring and then some radial nerves that extend out in the arms, but no brain, no centralization of function, which is, again, kind of a little bit of a throwback, right? Because insects have brains. You saw the brain of the crayfish. So we come to these guys, and they've actually kind of gotten rid of the brain, or at least a true brain. They have an endoskeleton now. Insects had an exoskeleton. They have an endoskeleton with an epidermis, all right, that actually covers this endoskeleton, composed of calcium carbonate, plates of calcium carbonate. I got a picture. I'll show you that in just a second. Ossicles, we call them. Again, calcium carbonate, folks, very common substance used by organisms in the sea. All right, sea stars, brittle stars, sea urchins, sand dollars, sea biscuits, and then sea cucumbers fit in this group. Let's take a look at the sea star a little bit closer. Specifically, let's look at this thing in the sea star called the water vascular system. Now, this is really very cool because the water vascular system is found in only one group of organisms. That's it. Nobody else has got them. There's no convergent evolution of any of this sort of stuff. These are the only critters that have this thing called the water vascular system. It's unique to echinoderms. So let me show you the way it works. What echinoderms use their water vascular system for is for movement and for capturing a prey. It's all driven by water pressure, right? So water moves in through something called a madreporite. You can see there in the picture. And the madreporite, the water then goes down this little thing called a stone canal. It's named that because it actually is very hard. It's like a stone. So it's a series of pipes into a ring canal, into radial canals, and those radial canals along either side have a bunch of these ampulla. Well, ampulla is a word that we use to refer to, I don't have one of my in the room here, too bad. The squeeze part of an eyedropper. That's the ampulla of the eyedropper. So these things kind of look like that, don't they? So what echinoderms do is they move water up and down these radial canals, in and out of these ampullae. And when the water moves in and out of the ampullae, it eliminates or decreases or it increases suction. All right, so if water is removed out of the ampullae and the ampullae shrink, it'd be like you squeezing the top of that medicine bottle. And when you squeeze the top of that medicine bottle and then it relaxes again, it creates a suction. All right, so what is going on there, you can see, like in this picture here, if they take water and force it out into the ampullae, so they increase that hydrostatic pressure, those ampullae will expand because of the water pressure. So this critter could be reaching out, right? Could be reaching out. So at the end of these ampullae, there are these little tube feet. And they can use those tube feet when they attach to something, and then they would draw the water out of the ampullae to create that suction. So these tube feet, if you look in the picture here, can actually stick to something. So they use these for walking along the bottom. Here you can see another picture of tube feet. Not all species have suckers. And as I said, they use them for movement. They're very thin, these tube feet, so gas exchange. Any thin surface in an organism, any thin membrane with a lot of vascularization, a lot of blood vessels is good for transmitting gases across the surface. They can also use them for feeding. What these organisms will do, sea stars like to feed on, say, for example, bivalves. And what they'll do is they'll wrap their arms around a bivalve. Now, remember with the bivalves in the clam, they have those two big adductor muscles, right? Adduction to pull together. So you got those two big muscles that hold that clam shell together. Along comes Mr. Sea Star, and Mr. Sea Star would like to eat the clam. Well, the clam doesn't want to be eaten, so it closes its shell. All right, so we got a closed clam shell here. The sea star wraps its arms around the clam shell, and it creates the suction in its tube feet and attaches itself to either side of the valves. So part of the sea star is attached to this valve, and part of the sea star is attached to this valve. And then they just very slowly, but very persistently pull. So they're pulling the shells apart. Now, if you're ever trying to shuck oysters, you know that it's difficult to get the shell apart. Some of that has to do with the fact that once they're dead, the muscle's a little tighter. But even if they're alive, it's hard to get that shell apart. So it's a battle between being able to keep my shell closed so the sea star doesn't eat me, and of course the sea star wants the food. Well, if you take those tube feet and you attach them to those valves, those shells, and you pull, you don't got to pull hard necessarily, but if you pull for a long period of time, what happens to muscles if they're being used for a very long period of time? What happens to muscles in general if they, if you were lifting weights for a long period of time, what would eventually happen to your muscles? Yeah, they would fatigue, wouldn't they? So the adductor muscles of those clams begin to fatigue. Now the shell begins to open just a little bit. You think, well, heck, it's got a long way to go, right? You got to open the shell all the way up. Oh, no, 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 no. The sea stars had figured out this clever way. They can actually evert their stomach. Their stomach's on the bottom side. Sea stars and many other echinoderms have an oral surface on the bottom where the mouth is. And then the other surface, the top, is called the aboral, the opposite of oral. So they evert their mouth from the oral surface. They can evert, that means stick it out of their stomach, out of their mouth, and then into the little crack of the bivalve, into that little tiny opening that those arms have pulled apart, and that stomach then can get down into the visceral mass of that bivalve, and the enzymes in the stomach can begin to digest all of that and liquefy it. And of course, as that happens, the organism's dying, and eventually sea star can open it all up and have its food. It must be very horrific for a bivalve, though. Can you imagine? It's like under siege. Slowly, day by day, my shell opens by a little bit, half a millimeter, and all it's got to get to is, just a small crack, and I'm history. So nature's kind of rough out there sometimes, isn't it? Here's what I mean by the ossicles, the calcium carbonate ossicles. These make up the bones, if you will, of the endoskeleton. They're called spiny skin. Some of those calcium carbonate materials made into a spine. That's good for protection, right? That's good for predators, keeping predators away. And oftentimes there'll be small little lumps, which are dermal branchia. Branchia means lungs. They can exchange gases across these, so oxygen can diffuse in and CO2 can diffuse out. Again, like I was saying, any highly vascularized surface, does a really good job of diffusing gases across it. All right, so the anatomy of a sea star, we have, as I said, there's the stomach. The anus is on the aboral surface, so it's on the top. The mouth is on the bottom. On the aboral surface, there's also, and I'll show you this in lab, there's a little madreporite. It's a little area that looks a little different than anything else, and that's where the water pours in for the water vascular system. The gonads and the digestive glands extend down into the radial arms with, again, the rest of the water vascular system as well. Now, in some organisms, the actual tube feet at the end, the podium, the part that sticks, actually secretes an adhesive and a dehiscent chemical. So they can actually not only use suction, but also stick to something, and then they can use a chemical to actually unstick themselves. These are mostly sexual reproducers, although they can engage in asexual reproduction. They can actually regenerate lost parts, like you see that a starfish over here. What needs to be there for that to occur, by the way, is you do have to have a piece of the central disc. So you can take a sea star and one arm, this is the one arm of the original, will generate into another one as long as you have a piece of this central disc in there. So it couldn't be cut off here. See what I mean? You have to be cut off a little bit higher. But this regeneration of lost parts is really fascinating, especially if you think about the medical implications of that. And on the right, that thing you're looking at with the white whatever coming out the top there, that's a sea cucumber. And what sea cucumbers will do is when you mess with sea cucumbers, they actually liquefy their internal organs, and then they spew them out of their mouth as this stringy, sort of sticky, silly string material. Predator avoidance. Predator's like, oh my God, this stuff's all over me. It leaves the sea cucumber away and alone, and then lives to another day. But what happens to their internal parts? Well, like I said, they can regenerate those. So they can regenerate those internal parts. Free-swimming larvae, yes, because we want dispersal, and gonorrhechism. So we have males and females of this group. Okay. All right, now among the echinoderm classes, we have the sea stars or the asteroidea. Sea stars are sea daisies. There's only 3 species of sea daisies that are known on Earth. This is one, Xyloplax jantae, yeah, discovered in 1986, living on the decaying part of old wooden ships in the bottom of the ocean. That's the only place they find it. It always makes you wonder what these things did, you know, before they were wooden ships. Now, I'm sure they existed, right? They existed because trees fall over into the ocean and, you know, they could live off of that. But my guess is when we started producing and sinking wooden ships, sea daisies may have found a boom in terms of the resources in their environment. Many times, members of this group, these sea stars, can be very important predators in a marine ecosystem. Like this here, this is the crown of thorns sea star. We call these kinds of organisms that are important in ecosystems keystone species. They don't have to be predators necessarily. The crown of thorn is a predator. A keystone species is a species in the environment that its presence or absence causes the biggest change in the environment. All right, so the crown of thorn sea star is a highly voracious predator. And when it moves into ecosystems, it causes massive degradation of the ecosystem by just eating lots of things and killing off lots of organisms. The keystone word comes from engineering. You know, when they built arches, see these all over. Oh man, I remember all those old churches in Germany, how wonderful it was to walk through those things that were 800, 1,000 years old. It's just incredible. I remember climbing to the top of the steeple in Ulm, Germany, and just thinking, my goodness, the people who hung outside and built this thing, you know, 800 or 1,000 years ago. The keystone is the block at the top, right? The idea behind an arch is the pressure. The blocks aren't glued together. They put pressure on the keystone. The keystone's the last thing you insert. The keystone species idea comes from if we remove the keystone, the arch falls, right? So the keystone species is going to be the one that has the biggest effect. Why can't I get rid of all that blue highlight? Anyway. Now, the thing about sea stars that separates them from another group that we'll move to next, which are the brittle stars, is the sea stars, the central disc and the arms kind of blend into one another. They're not sharply demarcated. And the arms taper. It's more obvious the way the arms taper. Don't get fooled by the multiples of 5, though, in that pentaradial symmetry. Not all of them have just 5 arms, like that one over there. What's that, 9? 1, 2, 3, 4, 5, 6, 7. Yeah, 9 arms on that Florida one, that Florida species here. Sea daisies aren't very big either, by the way. Look at that. They're less than a centimeter across. This guy looks pretty rough there, man. Looks pretty tough critter. Now the brittle stars. The brittle stars look like sea stars, but now the central disc is very obvious, and the arms don't taper as much. They're more regular, the same length all the way down, except maybe tapering at the end. They have tube feet, but they don't have suction cups. So they use their tube feet not so much for feeding, but for crawling around. They can actually move out on land. People get freaked out sometimes. I've seen them on videos. You can see these things actually moving across the sand. By using their tube feet and just kind of hooking them, kind of like the worms do with the setae, the chitinous structures on their body wall. They can hook these tube feet and they kind of move in through the dirt. The arms are easily autonomized. It's a really tough word because there's 2 T's in there. Automatized. Automatized means that they give them up. See the one in the lower right, the Amphiodia urtica. Under predator duress. This is what lizards do with their tail. Same thing. Under predator duress, that brittle star, the reason why they're called brittle, is because they will break off their arms with the idea that the arm's wiggling and hopefully the predator will mess with that. It's the same thing with the lizards, folks. When lizards are attacked, if the tail falls off and it's wiggling around, they're hoping that the predator will go, oh, what's this wiggly thing? And go after it, and then the lizard can crawl off and hopefully be safe. The picture in the background is a picture of the bottom of the ocean. We find a lot of brittle stars down at great ocean depths. The sea urchins or the echinoidea, the sea urchins and the sand dollars and the sea biscuits, they don't have arms. They do have tube feet, which if you look in the test, they have double rows of tube feet. The part here, see that lighter color? those are where the tube feet stick out through. The other parts where the spines stick out. So in this one that's really pretty blue colored one, the tube feet would stick out through here, and the rest of these are the spines, the protective spines. And movable, they can move them around. Obviously, that's good predator protection. To me, one of the most fascinating things about the echinoderms, or excuse me, the echinoidea of the echinoderms is a thing called Aristotle's lantern. This is their feeding mechanism. If you ever take a sand dollar or a sea biscuit and you shake it, you hear this rattling around inside. And if you open it up, you might see something like, oh, it looks like another little star inside. Well, sea urchins and sand dollars and sea biscuits have their mouths on the bottom, just like the sand dollar, or just like sea stars do. And the reason why we call it Aristotle's lantern, by the way, is because this drawing over here that you see was drawn by Aristotle, who was a brilliant naturalist for whatever his thoughts on the ladder of life. He was brilliant naturalist, brilliant scientist in general. It's a really cool thing, really precise mechanism. It has these calcareous plates that are connected to these teeth, and then these series of muscles, these protractor and retractor muscles that pull this way and pull this way. And it's like a mechanical lever. When those muscles pull back and forth, this thing pivots, and the pivoting of that causes the teeth to pivot. So if this is the calcareous plate and the teeth are at the bottom, as the muscles move the calcareous plate, the teeth pivot. And so when you look at the picture on the right, on the bottom, the teeth are opening and closing, and they're nibbling. They're grazers for the most part. Nobody else has anything like this as Aristotle's lantern. I just think that the whole way it's put together is just amazing to me. And it never stops to amaze me how in biology or in life these very intricate structures can be put together like that. Now, a couple of other groups that belong here with the echinoderms that really don't look anything like them are the things called the Crinoidea. These are the sea lilies or the feather stars. Sometimes they're called sea pens. We call these living fossils, again, for the fact that in this case, they don't look, well, they do, obviously, but the reason why they're a living fossil isn't because they look like a fossilized organism from a long time ago, but because they haven't changed a whole lot since the Cambrian, 500 million years ago. So this fossil of a Crinoidea right there looks very much like the modern day remnant, modern day descendants of that one. Now these are sessile critters that use their basically use their tube feet for filter feeding. They do have a free swimming larvae. They have actually sort of an interesting life, though. They have a free swimming larvae that becomes a sessile juvenile and then becomes a sessile adult. So the larvae settles down as a juvenile. Many times the larvae become an adult, then they settle down. But this one settles down as a sessile juvenile and then moves into a sessile adult. Feather stars, some of them are free moving. That's what you see. That's what you see crawling up on the beach there in the lower left. That's a feather star. It's going to be weird to see something like that. Again, suspension feeders. So in this case, the mouth is going to face up, and the anus actually faces up. But luckily, they're not like the flatworms whose mouth, or the cnidarians whose mouth and anus is the same thing. They have a U-shaped gut. So the mouth is one side and the anus is in the other. So they might be right next to one another, but at least they're not the same place. And it's not like the snail. Remember the snail, the anus dumped down over its head. That's what we call fouling. So, all right. Okay, so that, the sea cucumbers. Yeah, we can't forget them. Very bizarre. Common in the deep ocean, a reduced, very soft-bodied organism, so not much of an endoskeleton. And the the tube feet, the ones around the oral area of the body, there's some tube feet right there. They become feeding tentacles. So that's what the tube feet have become in these guys. They're important detritivores, which means they eat the decaying material, detritus, and we eat them. This is, I don't know what this is called, but this is what I found online for a dish of sea cucumber. No idea what it tastes like, but it would be interesting. They can actually respire through their anus, through these things called respiratory trees. Again, just a highly vascularized surface that they use to exchange gases. And there's this trick they use again. I remember a buddy of mine and I, diving in the Philippines one time, came across a sea cucumber. I'm going to switch slides here to chapter 34 while I finish this story. And him and I were picking it up and looking at it and just messing with it, and it started doing that, started spewing all this out. My buddy was, oh my God, it's underwater. It gets all over you, and it's kind of sticky. It does exactly what, it doesn't hurt, doesn't cause any pain or anything like that, but it does exactly what it's supposed to do. It gets you going, oh, you know, it gets you distracted from hopefully whatever you were trying to eat, or hopefully from being eaten. All righty, folks, chapter 34, last chapter. Here we are. We're going to be talking about the origin, the evolution of the vertebrates. And you see one in the fossil record quite clearly. What we got here is a spinal column. So this is going to be the big difference that we see in these critters. All right, so on the cladogram, there's our closest relative, the echinoderm. Us and the echinoderms in a monophyletic group descended from some ancestral deuterostome. And then having done the cladograms in class, you recognize what all these other things are. The notochord, the vertebra, these are all characteristics, right? All characteristics that arose. Now, over here we have a cephalochordate. We have urochordates. Cephalochordates and urochordates are those ones I was talking about. Cephalochordates are chordates, but they don't have a spinal column. So all those with a spinal column, the fish, amphibians, reptiles, us, all of us with a spinal column are chordates, but not all chordates have a spinal column. And again, a little bit more detail on that later on, but I know that's a bit confusing. The first vertebrates, again, it wasn't like this progression of organisms occurred about the time of the Cambrian explosion. So again, around so a lot of stuff went on back then during the Cambrian explosion. Out of the ocean too, by the way. That's a lot of life, for the most part, involved out of the ocean. First we get the fishes. First they were jawless, but when the jaws came along, whoa, man, when jaws came along, we talked about that in the Cambrian explosion. That really drove adaptation, because now you got jaws, you got some pretty significant predators out there, and you're going to have to become pretty good at getting away or defending yourself against the predator, and then of course the predator is going to have to stay pretty good at catching the prey. And then in a nutshell, so we got the fish, jawless at first, then they have jaws, then we get the amphibians, and they were the ones that reproduced in water but made it out onto land. They couldn't stay on land. Reptiles could live on land. Reptiles could live on land. They could lay eggs on land. They didn't have to go back to the water to lay eggs. So they became dominant. And that's, of course, you know, that's where the dinosaurs, that explains where their prominence. And then, of course, the dinosaurs go extinct, and then the mammals and the birds. The birds are not included in this group because remember now I said in the new science, birds are basically reptiles. We used to have another group in here we list as birds, but now we just accept the fact that birds are indeed just reptiles. So let's look at some of the characteristics that evolved. A notochord is a spinal column, not a spinal cord, so it would be the vertebral part of our backbone. Then there is true vertebrae. A notochord is a stiffened rod. It's not made of bone, but now we have a stiffened rod made of bone. Jaws, as I talked about, the mineralization of the skeleton, either through cartilage, if you're shark, or through bone, if you're, the rest of us, that made the skeleton stronger. Lungs, gills, lung derivatives, lobed fins. You see, the ability to get out on land, the ability to live on land started back in fish. There were certain groups of fish that in evolutionary history, there are certain groups of fish even alive today that have fins a little different than their counterparts. They're not the regular fish limb. They're lobe-finned. They're like if you were to put a flipper on the end of your hand, that's kind of what a lobe-finned fish is. And we'll talk more about those lobe-finned fish when we get into the fish, but that's a very important organism because, of course, lobed fins became limbs. And then limbs with digits. The amniotic egg. An amniotic egg is one that can be laid out of water. When a young is developing in an egg, they develop inside of an amniotic sac. So here's the embryo. There's an amniotic sac around it with fluid that keeps the embryo from drying out. Now, amphibians don't have an amniotic egg. They have an amniotic egg. So just put A in front of that word. Their egg doesn't have an amnion. The rest of us do. That's why we can lay our eggs without using water. The reason why amphibians lay their eggs in water, so they don't dehydrate. But with an amnion, you've got your self-contained water bath with you. So now you can lay eggs anywhere, by the way. You don't need to be near the water. The reptiles can move out into drier areas in the world. Remember at the time, that's what was happening. The interior of the continents were getting drier. So the dinosaurs were starting to rule. And of course, mammals, we also have amniotic eggs. The big thing about mammals, too, of course, where our name comes from is the fact that we nurture our young very effectively through the production of something called milk, right? Nobody else makes milk other than us. All right, the phylum Chordata, cord or string. As I mentioned, the nearest relatives were the echinoderms. Now, here's where we help fill in this idea of how an organism can be a chordate without a backbone. To be a member of the chordates, to be a member of the phylum Chordata, just like any other phylum we've talked about since even back in Bio 1, you got to share characteristics. If you're in the phylum Platyhelminthes, you share characteristics of that phylum. If you're in the phylum Echinodermata, you share characteristics of that. If you're in the phylum Cnidaria, the jellyfish, you share the stinging cells. That's one of the characteristics you share. So all chordates, and in parentheses, read that, that's very careful, at least at some time during their life, all chordates have a dorsal nerve cord. I say dorsal because that's unique to vertebrates. Remember when we were talking about the worms, they have a ventral nerve cord. That's why we know we didn't come from the worms. Too much to think, not very parsimonious to imagine the nerve cord flipped from ventral to dorsal. We have a notochord at least sometime during our life. Now, our notochord, us humans, is replaced later on by the vertebral column. Now, if you were a shark, that would be replaced by a vertebral column made up of cartilaginous material. And if you were the rest of us, that would be material that's replaced by bony material. Now, just because it's cartilage, as you'll see when you do the shark dissection next week, doesn't mean it's not tough. Cartilage is very stiff. It's functional as bone, okay? In you and I, we do have the remnants of a notochord in the adult. Those would be the intervertebral discs. If you slip a disc or rupture a disc or herniate a disc, that disc is the remnants of your notochord. You see, if you look down here on the left, this critter that we'll look at in a little bit more detail later on, this is a very primitive chordate. There's the notochord. The notochord is a stiffening rod. It's not made of bone. It's not the nervous part. You see, there's also a dorsal hollow nerve cord in yellow, but the notochord is the backbone of that. So this is what I mean about an organism that doesn't have a backbone, but is still a chordate. All right, because our backbone was developed from our notochord. But in some critters that are alive today, they still retain the notochord. All right. Now, gill slits. We all have gill slits. If you look in the mirror underneath your jaws, you can see the little gill slits, can't you? Well, no, of course not. We don't have gill slits or pharyngeal gill slits. But at some time we did. You see these pictures on the right? I'm trying to show you the similarity, like the chicken embryo and the human embryo. Look, pharyngeal pouches. Those are gill slits, by the way. Somites. Somites are the regular block formation of the muscles. The segmentation, that's segmentation. Look up above, fish, salamander, tortoise, chick. All of us start off looking kind of the same, right? We all have that dorsal nerve cord, a notochord, pharyngeal gill slits. It's just in the case of the humans, our pharyngeal gill slits don't end up as gills, like they do in a fish. They end up as the nerves and the musculature of the neck and the eustachian tube. You know, that tube that connects your inner ear to the rest of your to your throat, so you can balance out the pressure in your ear. And finally, a post-anal tail, right? Of course, our post-anal tail gets reabsorbed, and we're born with just a couple of bones down in the coccyx. And then if you were something like Cooper, my dog, he, of course, has a full-blown tail. All right, the somites, excuse me, I mentioned the somites. They gave rise to things like the muscle, the axial skeleton. The axial skeleton is the main skeleton, the spinal cord. The appendicular skeleton would be the arms and legs, that sort of stuff. So in this group, who we're going to talk about? Well, man, we're going to talk about a lot of critters. First, we're going to talk about a couple of critters that are very non-backbone-ish looking, like tunicates and lanceolates. And then we'll move on to the more traditional groups, the fish and the amphibians and reptile birds. Just put that in one group. And, of course, then we'll finish with the mammals. All right, now let's go back for just a second to this whole echinoderm larval form and being the ancestor of chordates. All right, so this is a little bit, well, not a little bit. This is a review of that patagenesis and patamorphosis. All right, I remind you once again in the upper left, there is the echinoderm larvae. It's bilateral. Right, it's bilateral. Now, we think what happened is through some evolutionary changes, through some modifications, through some, all those things we've talked about in this class all semester long, that that a bilateral echinoderm ancestral larvae developed that may have looked maybe something like this. Thank you, Pen, for not drawing. Maybe something like a little tadpole. Okay. And that tadpole, in some chordates that we're actually going to look at today, that tadpole is actually part of the reproductive cycle of these organisms we call ascidians. This is an adult ascidian. An ascidian is a tunicate. We'll get to the tunicates here in just a second, but this is a sessile organism. The tadpole larvae would be for dispersal. Call it a tadpole larvae because it looks like a tadpole, but of course it's not a frog. That picture up above, this really colorful thing, looks like a bunch of lamps. Those are all tunicates. Those are all ascidians. Their larval form looks like a tadpole. So the connection between the echinoderms and the chordates runs along this tadpole larvae. You see, if you remember back, this whole idea of progenesis, where you develop sexual maturity in a larval form, the question mark there says the hypothesis suggests that a tadpole larvae or a bilateral larvae of some echinoderm that was already around, that was involved in this tunicate reproduction, or perhaps it was another type of bilateral larva, underwent this patamorphic change, right? This patamorphic change whereby, as a larvae, it developed the ability to reproduce. Okay, so now I'm really using broad brushstrokes, but you get the idea. This is some of the earliest fish. This is is that an ostracoderm or a placoderm? I can't remember. This is an early fish. So the idea is, if patagenesis and patamorphosis and progenesis occurred, that some sort of a tadpole larvae could have developed the ability to reproduce, right, become sexually mature while it still looked like the larval form. And that larval form just so happens to look fish-like. And in some of the ostracoderms, or if that's a placoderm, I think those are ostracoderms. Ostracoderms have gone extinct. They're no longer around, but these eventually evolved into the other fish, and then fish into the amphibians, and so far. This is something we call Garstang's hypothesis. Garstang's hypothesis is a hypothesis to explain how the connection between the echinoderm group and then the chordate group. That, again, it wasn't a matter of an adult echinoderm. It was back at that developmental larval stage. You remember when we talked about developmental genes, right? Little tiny changes early on can have very profound effects, right? So if you make little tiny changes in when this organism is going to become reproductively mature, if you speed that process up, you'll get something that looks like a fish or a tadpole, kind of like larval, that is actually a full-grown adult organism, can reproduce itself. All right, so now let's start looking at the different groups. Let's start with the first group, which we call the Cephalochordata. All right, so now the Cephalochordata, the word, if you look at the cephalochordate, basically cephalochordatas are chordates with a head. And you'll notice that one of the characteristics to be in the chordate group, of course, is that you have a notochord. Now, cephalochordates have, or had, and still have a notochord. We have some of these in lab, but you see in the picture on the finger there, somebody's holding what is called an amphioxus. Amphioxus. Let me type that word in. There's lanceolate is the kind of the common name. Amphioxus is a scientific name. Amphioxus, and then so is branchiostoma down here at the very bottom. I think Amphioxus is the older name. I remember right now. It's Branchiostoma. Little tiny critters, no bigger than a few inches long, right? Scaleless, look, persistent notochord, all right? Dorsal nerve cord, okay? Pharyngeal slits. Water moves in through the oral hood and the tentacles across the pharyngeal slits where oxygenation occurs. Then it dumps into an atrium or an open area, and then it goes out. The water goes out through the atriopore. So water pours out through the atriopore. Of course, the digestive material pours out through the anus. So we got pharyngeal slits, dorsal nerve cord. We have a notochord, postanal tail. So you're a chordate. You may not have a backbone. You have a persistent notochord. So to kind of make it easy on myself, what I've done in the upper right is I've listed the 3 groups of chordates, the big groups we're going to talk about. Now, the vertebrata is the birds and the mammals and all those other ones. But these 2 special groups, the first one I just introduced you, the cephalochordates, and the next one, the urochordates, I like to call them the non-vertebrate vertebrates. So instead of calling them the invertebrates, which would be wrong because they're not invertebrates, I just kind of changed it and said, okay, these are non-vertebrate vertebrates. They are chordates, but they don't have a vertebral column. Same thing with the urochordates, all right? Urochordates are the tunicates I was talking about. The word tunicate comes from very often tunicates are wrapped in this sac, many times made of cellulose, a cellulose sac. There's some interesting convergent evolution, isn't it? All right, a sac wrapped in cellulose, just like we see in plant cells. The larval form of a urochordate, there's one. Look at that, tadpole. Looks like a tadpole, doesn't it? All right, so we're in that, we're in that Garstang's hypothesis that maybe it was some tadpole that metamorphosed into a free-living adult organism. Now, the urochordates are around today. You can find them growing very often. They're sessile. Very often see them growing on piers and stuff. We call them sea squirts sometimes. You may not even know that it's an animal. You grab it and squeeze the water out of it is where the sea squirt comes from. But they're filter feeders. Water flows into an incurrent siphon, kind of like the clam, right? Out through the excurrent siphon. The pharynx, of course, with all these numerous slits, is used for oxygenation and food gathering. And there's the tunic, the outer covering. The tadpole is free swimming, only lasts a little bit of time. It doesn't even feed. So it's going to disperse and then immediately settle down and start changing from a tunicate larvae to an adult tunicate. All right, so there's the larvae on the left, kind of fish-like, to the adult tunicate. Looks nothing like a fish. The adults are sessile, right? Immobile filter feeders. Only in the ocean. You only find them in the ocean. Tunicates, they come in all kinds of different colors and bizarre arrangement. Here's another tunicate. This is a salp. A salp is a member of this group. Now, what's interesting about a salp, it's an ocean critter. I think I talked about this in marine science. I think I talked about salps. It's an ocean organism that is it traps food in a mucus layer. This is all mucus. The actual salp organism is just that right there. So the salp organism produces this mucus, and then as it's floating through the water, food sticks to the mucus, and there you go. I got myself a meal. All right, so that, along with, like I said, the cephalochordates make the non-vertebrate vertebrates. So now we're actually ready to move into the vertebrate vertebrates, the rest of the group right here. Okay, now, where did we go from the non-vertebrate vertebrates to the vertebrates? Well, increase the complexity of the nervous system. Organisms got bigger and they got more sophisticated. We developed the skull. It goes right in with the development of the nervous system. Because if you develop the nervous system, you're also developing the brain. And if you're developing the brain, you're going to want to protect that brain. So one of the things we get is this development of the skull. In aquatic organisms, we get fins, which makes sense because they're going to live in the water. But evolutionarily speaking, the significance of the fins was where the legs came from, so that organisms that crawl down the land, obviously it's real hard to walk on land with fins. Legs are much better. Overall, what we see among the vertebrates is much more increased efficiency in things like food capture, if you're a predator, as well as predator avoidance. So arms races, like predator and prey things, drive evolution at a very fast pace. There's nothing like a very efficient predator to put a very distinctive evolutionary pressure on a group of prey to survive, or at least some of them to have some advantageous variation in which they can survive. Okay, so now we're in the vertebrate groups. Let's take a look at these groups up here. These are fishes, all right? By definition, we call them fishes. Of course, sharks are also fish. These are typical fish, ray fin fish, and then the lobe fin and lungfish. So there's a lots of different kind of fish. Let's talk about the Myxinae first. This is a Myxinae. They're not the most attractive, or come on, click for me. There we go. They're not the most attractive organism in the world. Myxinae are jawless creatures with a persistent notochord. So here we are even into the vertebrae, what we call true vertebrates. And what we have here is an organism that still doesn't have a vertebral column and has a notochord. Again, if you're going, oh, I'm confused, Aristotle's ladder, folks. You can't go from the non-vertebrate chordates, the cephalochordate, the tunicates, immediately to the vertebrate chordates. You're going to have some of those vertebrate chordates that are a little bit more than the non-vertebrate chordates, but not quite. They do have a skeleton, all right? They have a skeleton made of cartilage. They have a skull, not much of a brain. These are very often parasitic organisms, and in many times parasites don't need to have really well-developed brains. They do have eyes and ears. They have teeth made of keratin, which is the keratin is your nails are keratin, so it's very hard. It's good for making teeth. If eel skin wallets, purses, anything called eel skin is actually not made out of eel. This is not an eel, all right. This is actually what we call a hagfish. Such a lovely name, hagfish. So I guess probably from a marketing point of view, it makes more sense to call that wallet a eel skin wallet than a hag skin wallet. Probably wouldn't sell very well, but that's what that stuff is. It's not eel. Eel is a more proper fish. These things are fish-ish. They are fish, but not quite like the eel. All found in the marine. Slime eels, too. They produce copious amounts of slime. Oh, you can see the slime glands. Slime is really good for an organism that lives down in the substrate, in the rocks, in the sand. It's also good if you're an organism that might engage in battle with other organisms as you're trying to parasitize them. It's a protective coating. I can't remember if I said in y'all's class, or maybe it was the other class. I came up with this idea that, like, boxers will put Vaseline on their face before a fight. It's a protective coating. It's the same thing that the slime is for these guys. So the slime eels, not really eels. Eel by proper is another type of fish. Now the slime eels and the next group, the hagfish, we're going to talk about, all have round mouths, so they're called the cyclostomes. They have a round mouth as a cyclostome. Stome is mouth. So it's these guys and these guys, the lampreys. These are the lampreys, Petromyzontida. Okay, most of these are parasitic, and some are invasive. Like, for example, Petromyzon marinus, which is the sea lamprey, but can actually live in brackish and fresh water, has made its ways into the Great Lakes regions. Oftentimes probably because of that whole idea I talked about with the zebra mussel, right? The larvae being carried in the holds of the ship, so as the ships move into the Great Lakes region, or they maybe just, in some cases, swam up the river there, the St. Lawrence Seaway. They're scaleless, cartilaginous skeleton. They have a notochord still, so we don't have this bony structure yet, but they are developing lateral neural cartilages, as you can see in the picture, these little blue things. And if you've ever seen a picture of your vertebrae, the vertebrae of other vertebrates, you got the round part, so this is where the disc would be. That would be the remnants of your notochord. And then there is an arch at the top that the spinal cord, the nerve cord, goes through. Right, that's called the neural arch. It protects the spinal cord. We see the beginnings of that in these lampreys with these lateral neural cartridges. Round mouth, teeth of keratin, just like in the hagfish. Rasping teeth. This is the mouth of this organism. The way they live, these are gill openings, by the way, in the eye. The way they live is they attach themselves to a host fish, like you see here, and then they use these keratinous teeth in that round mouth to actually rasp a hole or chew away a hole in the outside of the fish. And then they swim into the fish and they eat the guts out of the fish from the inside out. I remember when I was in grad school out fishing, and we would pull up a big fish, and when we would cut it open, it would have lamprey inside, because the lampreys would have already chewed their way through and actually digested the internal organs of that fish. Lovely way to live, right? I've only got a couple of minutes, so I just want to start this group because we're going to be moving into the fish. I got a lot that I want to talk about in the fish, but we're going to move now from the cyclostomes, the round mouth, to the gnathostomes. All right, so these are the organisms that have the regular mouth with the jaws, right, the upper and lower jaws. And so what we'll do when we get next time is we'll get into the cartilaginous fish, the sharks, the bony fish, the lobe fin fish, the lungfish. So you see, we've got a lot of fish to talk about before we actually get to the terrestrial critters. All right, so that's all I'm going to do for today. I'm going to stop the recording. If you have any questions, by all means,